Hello, my name is Matthew Hahn from Indiana University. My talk title is The Ortholog Conjecture Revisited, the value of orthologs and paralogs in function prediction. I'll be talking today for my co-authors, Moses Stambulian, Rafael Guerrero, and Peja Radovoyets. As of today, 2 weeks before the talk, I will not be present for the Q&A. Peja will be. So I'm going to talk about the need for function prediction. So why do we need to predict function? We need to predict function because there are so many sequences being collected. This is a commonly viewed graph. Lots of sequences because the price is dropping so much, but very few experimental annotations. Just as an example of this, the number of vertebrate genomes in the ensemble database alone is over 200. The number of these vertebrates that are in a lab with people actually doing experiments on proteins from them is probably not more than a dozen or 2 dozen. So we need to predict function. The way this is normally done is by transferring annotations. We transfer annotations from a protein with experimentally annotated functions. We transfer them to proteins with fewer or no annotated functions. Now, there's a whole research field of how exactly to choose which proteins to transfer the functions from and to. I'm just going to say that one commonly used way to do this is by using evolutionary relationships among these proteins. So there's lots of different types of evolutionary relationships. The most common types that people distinguish among for this purpose are orthologs and paralogs. So in this tree, there are 3 species, A, B, and C. Some of the species have multiple copies of different genes. Orthologs are related by speciation events. So in this example, A1 and B1 are orthologous genes because their most recent common ancestor is a speciation event. The same is true of A1, B1, and C1. Paralogs are genes whose most recent common ancestor is a duplication event. So as you can see, C1 is a paralog of A2 and B2. It's also a paralog of C2. If all duplication events were older than speciation events, it would be really easy to choose which proteins to use for function prediction, but that's not always the case. Sometimes there can be very closely related paralogs, as with A1 and A3 shown here. But a guiding light in how to choose which evolutionary relationships to use in function transfer is what we call the Ortholog Conjecture. The Ortholog Conjecture states that orthologs should be strongly preferred in function prediction because they evolve functions more slowly than paralogs. So which evolutionary relationships should you use? You should use orthologs. Now, this is not a necessary condition. There are some methods that do very well without respect to using orthologs or paralogs. They don't have to use one or the other. For instance, Sifter by Barbara Engelhardt and colleagues uses both, and it does very well in all function prediction tasks. But in general, the field of function prediction by evolutionary relationships has used the ortholog conjecture. So orthologs should be strongly preferred. But it's not really a quantitative kind of conjecture. It doesn't say, well, orthologs should be preferred by so much, but for instance, if there is a paralog that's more closely related, you can use that one. No, instead it just says, well, we should only really use orthologs. So I think in practice, the way the ortholog conjecture has been used is that orthologs should be the only kinds of proteins used in function prediction because they evolve functions more slowly than paralogs. I should say that it's uncontroversial that paralogs evolve by sequences. Their sequences evolve faster than orthologs, but there's much less data about the evolution of function. Just as an example of the focus on orthologs in the function prediction field, here's a screenshot from the Quest for Orthologs group, a group of very good scientists who are trying to identify orthologs. The text there says proteins and functional modules are evolutionary conserved, and this conservation allows knowledge transfer between well-characterized model organisms. As we've said, the second part says the underlying biological concept is called orthology, and the identification of gene relationships is the basis for comparative studies. So now the idea of functional transfer has become inextricably intertwined with the idea of orthology, when, of course, that's not necessarily the case. So almost 10 years ago, we set out to test the ortholog conjecture. So are orthologs really more functionally conserved than paralogs? To do this, we took human proteins with experimentally annotated functions. We took mouse proteins with experimentally annotated functions so that we could compare how well the functions were in common between these two organisms. We use these two because they both have a lot of experimental annotations and because we have good evolutionary relationships. We can recreate the evolutionary relationships among orthologs and paralogs quite accurately, so we can compare orthologs and paralogs between these two species. And here's what we found. We found that paralogs are more similar than orthologs. So along the x-axis here is the sequence identity, the amino acid identity between any two proteins that we're comparing. And on the y-axis is a measure of functional similarity, in this case, Maryland Bridge. In all of the graphs I'm going to show you, it doesn't matter which measure of similarity we use. I'm also showing you here only biological process. It also does not matter which ontology we use, biological process, molecular function, cellular component. None of the results I'm going to show are qualitatively dependent on any of those choices. So as you can see, paralogs are more similar, especially at higher sequence identities than orthologs. But we can break down paralogs into multiple different types of genes. If there's only two species, there's a convenient way to break them down into in-paralogs and out-paralogs. In-paralogs happened more recently than the speciation event. So here the speciation event between human and mouse. So H1 and H3 are in-paralogs, and out-paralogs happen more distantly in the past than the speciation event. And so we have within species out-paralogs like M1 and M2. They share a duplication event before the split of human and mouse, but they're both found in mouse and between species out-paralogs. Orthologs, of course, are always found between species. And what we find when we break genes down into these different categories is that all of the types of paralogs that are within the same species are more similar than orthologs. So within human or within mouse, at essentially all sequence identities. And again, it's true of biological process and every other, and molecular function, and for whatever measure of functional similarity. Those results that I just showed you were from across many different gene families. Some families had only orthologs, some families had only paralogs. So maybe there are some biases there. So we looked at only within families where we have a family that we can compare both paralogs and orthologs within the same family. When we do that, we find if there is a paralog with higher sequence identity, the paralog has higher functional similarity about 4 times as often as the ortholog does. If, on the other hand, there's an ortholog with higher sequence identity, we still find paralogs with higher functional similarity about twice as often. So that's probably driven by these within species paralogs. We can also, if we're uncomfortable with gene ontology and those annotations, we can also use gene expression profiles and the correlation across 25 homologous tissues in human and mouse to ask which are most similar. And again, paralogs, and especially within species paralogs, are the most similar. So here's a tentative model to explain the evolution of function, a model that we can think about as we go forward in the talk. The first idea that our data suggests is that function evolves at a rate proportional to time. Okay, so the rate of function evolution is not proportional to sequence evolution, but time. So that idea explains 2 important patterns that we observed in our data. One is that orthologs are flat. The relationship between functional similarity and sequence similarity is flat. And that's because all of the orthologs are exactly the same age. They have exactly the same amount of time back to their most recent common ancestor, no matter what the sequence identity is. It also explains why the paralogs do go down with age, and that the functional similarity goes down with age, and that's because paralogs can have duplicated at many different times in the past. The second thing we have to pay attention to is that it's clear that the cellular environment is key to function. Okay? Being in the same species is key to function, and that's true of biological process and molecular function. You may be an enzyme in whatever species you're in, but the type of enzyme you are and the types of reactions you catalyze differs depending on which species you take part in. Okay, so those were the main results 9 years ago, and boy, people had opinions about those results. And there were a lot of opinions, and I think a lot of good criticisms, some of which I'll cover. But really, people wanted to save orthologs. People thought orthologs should definitely be more functionally similar than paralogs. Here's a quote from one of these papers: "The orthology field was baffled by a publication claiming that orthologs are less functionally conserved than paralogs." And I will say that many of these papers did do new analyses, some of which showed that orthologs and paralogs were equivalent in function, some of which showed that orthologs were slightly more similar in function. But other weird things would happen in these analyses, like evolution would not, function would not evolve over time, or function would evolve over time such that more distantly related proteins became more similar. So there were problems. I should also say, of course, that there were papers that supported the results that we presented in that paper, showing that paralogs indeed were more similar than orthologs and are certainly, had lots of functions in common. So let me talk about some of the criticisms, because I do think, as I said, they're valuable to consider. So one problem with the original analysis is that Go annotations are not equivalent across species. And I think this is really key. So one reason is that they're species-specific terms. If I want to predict, let's say, a certain kind of cancer that only occurs in humans, well, I'm going to find that genes found in humans are better at predicting that function, right? So it's a little bit unfair to try to compare how these proteins evolved if I'm only using some terms in one of the two species. The other interesting thing is that researchers also tend to use first a subset of terms. They don't know the entire Go vocabulary, but they also tend to study genes in the same species. And so they're more likely to annotate other genes in the same genome. And that's a problem because missing annotations are interpreted in the types of functional similarity we're using here as differing functions. So for instance, if the human and mouse proteins shown here, the DAG from Go, are exactly the same, but the green function is only studied in humans, then we would say these proteins were different, simply because we don't have them annotated in mouse. We did try to control for this by using only proteins from different papers, and we still found paralogs were more similar, but it doesn't completely control for it. So a study that reanalyzed some of these data by taking out all shared authors, not just shared papers, and by trying to remove terms that were especially overrepresented within a species, found for human versus mouse that there was actually no difference between orthologs and paralogs, and that in two yeasts there was slightly more similarity between orthologs and paralogs, as the title say. And I mentioned the yeast too because I'm going to use those in our reanalysis. Okay, another problem with the original analysis is that there's simply maybe not enough Go annotations. And so I like this analysis. It turns out that the number of annotations was obviously accumulating over time, and it was accumulating such that orthologs were becoming more similar over time. And this paper has a nice prediction. They predicted that functional similarity in the three Go aspects, biological process, molecular function, and cellular component, would become greater than for orthologs and paralogs in 2013, 2018, and 2013. So that's a testable hypothesis. So here in today's talk, in the few minutes I have left, we're going to revisit these same ideas. And again, we're going to know about these possible biases, and we're going to have many, many more times data than we had 10 years ago. But I think more importantly, what we've realized now is that there's actually two separate ortholog conjectures. The one that we had posed in the 2011 paper, which is written here, is really an evolutionary conjecture because it's combining the pattern that orthologs are more functionally similar than paralogs with the process, which is that evolution is driving this. And I think we can simplify this to just this function prediction conjecture, which is orthologs should be strongly preferred because they're better predictors, regardless of the types of the reasons for this. And this is a really important distinction because the problems with the Go terms, the annotations that I've mentioned, only affect the evolutionary conjecture, right? So Go annotations aren't equivalent across species. People use different terms across species, but that doesn't affect the function prediction conjecture because all I want to do is have accurate function prediction. I don't really care what terms are used in the other species and whether authors tend to use one term more than the other. As long as I assume they're giving me accurate annotations, that's okay. So for instance, if I want to predict functions associated with mice tails, I probably don't want to use any studies from humans. I know I'll do better at studies from mouse, but I don't want to get rid of that. That's not a bias. That's part of my job. Okay, so two parts to testing the function prediction conjecture. First, which are more functionally similar, orthologs or paralogs? And second, even if we find that orthologs are more similar, they're only a little bit more similar. That's what all of the data suggests so far. So how much do we lose by excluding paralogs? Why should we be excluding paralogs at all? So again, we're going to use human mouse and the two yeasts, Saccharomyces cerevisiae and Saccharomyces palmbi. We're going to take experimental annotations from more than 80,000 papers. It's about 6 times more data than we had almost exactly 9 years before. And here's what we find. Again, in human and mouse, in paralogs, within species, paralogs are more similar than orthologs. Now I'm using Yang-Clark functional similarity, but again, the measure functional similarity does not matter. I can do the same analysis in the two yeasts, and again, it does not matter. I always find within species paralogs are better at predicting functions. They have more similar functions than between species proteins. Okay, so we can test the evolutionary ortholog conjecture a little bit. I've tried to remove all shared authors. We try to remove as many biases as we can. We still find that human and mouse are more similar in paralogs than they are in orthologs, and the same goes for the yeasts. Okay, so which are more functionally similar, orthologs and paralogs? Paralogs, definitely. So how much do we lose by excluding the paralogs? Even if paralogs are better, maybe there are just many fewer of them and they're more distantly related, and so they don't affect our predictions a lot. So we can quantify this. First, we can quantify how many genes do we lose. So here are all of the gene families, those are the top numbers, and the number of genes within them, and the types of categories they fall into. And I will say that if you follow the strict ortholog conjecture, you use only one-to-one orthologs, you would use only 4,700 pairs of genes. You would essentially ignore all of the other data. So you would ignore 75% of the data. There's 3 times more data that is not bound up in orthologs than is in just one-to-one orthologs. This is for human mouse. Similar thing goes for the two yeasts. There's many fewer duplicates, but you still lose about 50% of the data. Okay, so you lose lots of the data. How does that affect our predictions? So we can do an analysis where we ask how good are our predictions with all homologs, how good are our predictions if we remove orthologs, and how good are our predictions if we remove paralogs. So you can see here for both species comparisons, if we remove either orthologs or paralogs, our predictions suffer. And they suffer about equal amounts. So these are families with orthologs and any type of paralogs. If we restrict ourselves to families with orthologs and in paralogs, actually removing orthologs does not hurt our predictions. Only removing in paralogs does. So what this says is, if we have in paralogs, actually orthologs are not helping us. The in paralogs are giving us the most accuracy. So how much do we lose by excluding paralogs? A whole lot. So here are my take-home messages. There really are 2 ortholog conjectures. There's an evolutionary conjecture and a function prediction conjecture. The evolutionary conjecture is very hard to test. I think there are some exciting new technologies that might allow us to test it, but it's really hard to get around lots of the biases. So the function prediction conjecture, the ortholog conjecture is overwhelmingly rejected. Orthologs are not more functionally similar than paralogs. And finally, I think really what our data suggests is use all of the data, right? Use orthologs, use paralogs. There's no reason we should be excluding either. There are methods out there that allow us to use both. With that, I'd like to thank our funding agencies and thank you for listening.